Hello everybody, welcome to my channel. Myself, Dr. Renuka Joshi. Today I am going to explain about one of the important repair mechanism. Now, majorly we know that there are the different types of the damages which are occur within the DNA which are responsible for distorting the structure of the DNA. So, to restore the proper structure of the DNA within the prokaryotic cells, there is appearance of or there is presence of a various types of the repair mechanism. So, today we are going to discuss about one of these important type of the repair mechanism which is known as the nucleotide excision repair. Nucleotide excision repair. So, let us start with the nucleotide excision repair. Now, majorly within this nucleotide excision repair, there is the removal of the large lesions. Means whenever the DNA damage, it is in the form of large lesions. For example, majorly the thymine dimer. Means instead of the specific type of the modified bases, if we want to remove the thymine dimer from the DNA which are occur within the DNA as a result of the exposure of the bacterial cells to the UV radiations, such type of the thymine dimers, these may be get removed by the activity of this nucleotide excision repair. Within that, a specific type of the enzymes, these are get utilized which are known as the exonuclease, or majorly whenever we are considered this type of the mechanism, specifically in case of the Escherichia coli, which is considered to be a model organism during the genetic study of the different bacteria. Within this E. coli, this nucleotide excision repair, it is carried out with the help of UVR-ABC complex, means specifically a enzyme complex which is utilized for the repair of the damage which is occur due to an UV light which is acts as an mutagenic agent. This UVR-ABC complex, it cuts on the either side of the damage, that is on both side of the DNA damage. It is responsible for cut the DNA and excise an oligonucleotide, means a large DNA fragment which contains that DNA damaged region, it means the thymine dimer. The thymine dimer containing the fragment of DNA, it is get removed by the activity of this UVR-ABC complex. Now this nucleotide excision repair system, it involves actually the four different types of the enzymes, that is UVRA, UVRB, UVRC and the UVRD. This UVRD, it is also known as an DNA helicase enzyme. Now we are going towards what is the mechanism of this nucleotide excision repair. Now within the first step of this nucleotide excision repair or the NER system, there is an recognition of the damage. Means firstly it is essential that at which site the damage it is occur. So this recognition of the damage within the DNA, it is carried out by a heteromeric trimer. Heteromeric trimer means within that there is the involvement of two different enzyme complexes, that is the UVRA and the UVRB. And as it is an trimer, it means the three units these are involved, that is the two units of the UVRA, that is the dimer of UVRA which combines with a single unit of the UVRB, and combinedly these are responsible for forming this heteromeric trimer. And this heteromeric trimer, it is responsible for the recognition of the damage which is present within the DNA. Now in this nucleotide excision repair, it is highly energy consuming. Each step which is involved within this nucleotide excision repair, there is the requirement of energy. This energy, it is consumed in the form of hydrolysis of ATP. Means firstly for the formation of dimer of the UVRA, there is the ATP hydrolysis. Then for the purpose of the joining of the trimer, that is for the purpose of joining of the UVRB with the dimer of the UVRA, again there is the utilization of energy, that is the ATP hydrolysis, it is occur. In this way the two UVRA units, these are combined where there is the ATP hydrolysis, again there is the joining of the UVRB and again there is the hydrolysis of ATP. Now within the next step there is the removal, means after the recognition of UVR damaged region within the DNA strand, there is the removal of the dimer of UVRA, and only to that damaged region there is the binding of UVRB. B. Then this UVRB, it is get assisted by the another enzyme complex which is known as the UVRC. Now both of this UVRB and the UVRC, these are acts as an endonuclease enzyme. Means here there is an endonucleolytic cleavage, it is occur within the damaged DNA strand by the activity of UVRB and the UVRC. This UVRB complex, it is responsible for cleaving the phosphodiester backbone of the DNA to the 5' end of the damaged, that is from the 5 nucleotide away from the 5' end of that of the damaged region, and the UVRC, it is responsible for cleaves to the 3' end, that is 12 nucleotide away from the damaged site. Means total about the 12 to 13 nucleotide base pair region, it is get cut due to the activity of this UVRB and the UVRC. Now after the double cleavage which is carried out by the UVRB and the C, there is the detachment of the UVRB from the DNA, and then there is the recruitment of another enzyme complex which is involved within this nucleotide excision repair, which is known as the UVRD, that is nothing but the helicase enzyme. No doubt the endonucleolytic cleavage, it is carried out within the DNA by the UVRB and C complex, but due to the presence of the hydrogen bonding in between the complementary DNA strand, there is no removal of the damaged region from the DNA strand. So for that purpose, majorly there is the activity of this helicase enzyme, it is important. And after the activity of DNA helicase enzyme, that is the UVRD, a 12 to 13 nucleotide long DNA which contains the damaged site, that is for example it is, if suppose it is an thymine dimer, then the DNA segment which contains the thymine dimer, it is get removed from that DNA strand. Here within this figure we can observe how this reaction it can occur. So here it is an DNA region or it is an DNA strand, damaged DNA which contains the thymine dimer. To that, firstly there is the attachment of the UVRA complex, that is the dimer of UVRA complex along with the UVRB, that is the heteromeric trimer. It is responsible for attached with the DNA strand, and they are searching or scans the DNA for the damaged region, and they are fixed at that site where there is the damage, it is occur within the DNA strand. Now after that there is the, means after searching the damaged region, there is the removal of UVRA, and then this UVRB, it is get assisted by the UVRC. Now both of this UVRB and the UVRC, it is responsible for creating the cut on both sides of the damaged DNA, and after that the UVRD, it is get assisted. After the activity of UVRD, now there is the removal of UVRB and the UVRC, and there is the removal of the damaged region. Means the removal of damaged region, it is carried out by the combined activity of UVRB, UVRC and the UVRD, and there is the, after the removal of this or after the activity of UVRD, again there is also the removal of UVRB and the UVRC. Now after the removal of the damaged region from the DNA, the UVRC, it is also get released from the DNA, and now the gap which is formed within that DNA region due to the removal of the DNA fragment with the thymine dimer, this gap, it is filled by the another enzyme which is also participated within this nucleotide excision repair, which is nothing but the DNA polymerase first enzyme. As we know that like that of the DNA polymerase third, this DNA polymerase first enzyme, it also possesses the 5' to 3' polymerization activity, and with its this polymerization activity, it is responsible for filling the gap by considering the another undamaged strand as an template strand. At last, now no doubt the DNA polymerase first, it fills the gap, but a nick it is remain within that DNA strand. So to continue this strand, majorly the sealing it is important, and this gap sealing it is done by, or this nick it is sealed by the activity of the DNA ligase enzyme by forming the phosphodiester bond, and by this way there is the completion of nucleotide excision repair. Here within this figure again you can observe the same events that I am explaining here. Now here the DNA polymerase first, after the removal of gap, it fills the gap by using the 3' hydroxyl group, and it fills the gap this gap it is get filled, but a nick it is get remain within the DNA strand, and this nick it is get sealed by the activity of DNA ligase enzyme, and by this way there is the damage it is get repaired through the activity of nucleotide excision repair. So here it is a mechanism of nucleotide excision repair. If you like this video, please subscribe my channel and also share this video with your friends. Thank you.